Welcome to UT Brainstorms. Thank you for being here. This is our 14th Brainstorm, so thank you for that. We have a lot to be thankful for. I want to thank our A's, Elena and Alondra, who help organize this. They're out front, and their bevy of volunteers from UT and from St. Edward's. So thank you to them. Thank you, guys. Thank you to many of you. I know that how many of you are here because you heard about it from a friend yeah, so thank you for the friends for doing that, spreading the word via Facebook and what's the other way? Talking to people. So I'm getting old. So, and thank you to our supporters. We had a horn raiser, kind of UT's version of GoFundMe, and we raised some money this past fall. Thank you for that. Teresa Binning is here. She was our guardian angel who was the source of the match. So she matched every contribution that you guys made. So thank you to and thank you to Teresa. So I always like to start to find out a little bit about who's here. So how many of you are here for the first time? Who's first Brainstorms? So this is always interesting for me because on the one hand, it's so exciting to see so many new faces, and on the other hand, maybe we need to work harder to get people to come back. How many of you have been to maybe more than 5 or 6? Yeah, okay, we're still good. Okay. Thank you for being here. So. My name is Mauk. I'm a member of the Department of Neuroscience at UT and the Center for Learning and Memory. And Brainstorms is sponsored by the neuroscience community here at UT, and it's just our way of reaching out and starting a conversation with the Austin community. It's our belief that as a world-class neuroscience program, that one of the things that we should be doing is sharing what we know and our enthusiasm about neuroscience with the people who fund us and with the people who we live around. So it's always intended to be a conversation. So that means here we'll do our best to answer your questions. I'll try to inform you a little bit, but that's just a springboard for the questions. As always, the conversation needn't end here. You're always welcome to email me. Email me if your question didn't get answered. Email me if you thought of a question on the way home that you wished you'd asked, or email me at midnight when you think of that amazing question that only happens at those times. So we very much want this to always be a conversation and not just what happens right here with me talking. All right. So this afternoon I want to talk about Alzheimer's disease mostly, but it could really be about any of the many neurological diseases, the progressive neurological diseases. We'll focus on Alzheimer's disease. And the thing is there's so much that we could talk about. So there's so many things that we could talk about. We could dive into details of the latest theories about what causes Alzheimer's and how we might stop it. We could dive into all of the things that float around the internet about what you can do to prevent it, or what homeopathic cures, or this and that. We could talk about funding for Alzheimer's disease. We could talk about the progression of the symptoms and what to look out for. We'll talk about many of those things. But mostly what I want to do, because there's so many details and so much to talk about, I want to focus on some things that you could walk away with, some skills that you could walk away with from the standpoint that knowledge is power. And so what I mean by that then is this is what I'd like our goals to be tonight. So my hope is that you'll walk away a better consumer of all the things that you might read. Right? There's a lot out there. It's confusing. People make claims, hard to say. And so one of my hopes is that you'll just be a better informed, more agile consumer of the information that you would see. And part of that, frankly, is my hope that you could be better able to protect yourself from all the snake oil salesmen out there. There's a lot of them, unfortunately, and they confuse the issue, and this is part of what we want to talk about tonight. And this is the part that will take a little bit of work maybe to introduce to you, but I'd like you to be able to better imagine what a cure might look like when it arrives. And that might seem like a strange thing. As you might think, well, you scientists, you just do your experiments and you discover things and then the cure happens. But that's not exactly the best way for science to work. So much of science is about imagining what could be. And my favorite example of this is always Albert Einstein. He was a brilliant mathematician, but one of his great skills was his imagination. He would use thought experiments just to imagine how things might be. And the value there is that there's so many possible answers. There's so many avenues to pursue, so many things you might try. And using your imagination and imagining what the answer might look like is a powerful tool to narrow down the possibilities and make it digestible, make it possible to succeed. Einstein loved to imagine what it would be like to ride on a ray of light. That was his way of thinking about space and time and where special relativity came from. So this isn't physics, and I'm not Einstein, and nor are you, but we can make ground in that way, and I want to focus it in this way. And so this is just me because I like alliteration. But so I made this up, but I want to. This is what I mean by what a cure might look like. So we can imagine cure or a treatment taking different forms. So starting at the bottom, it would be wonderful to have the ability to prevent Alzheimer's disease, to stop it from happening in the first place. Now, if that's all we could do, then if it happened, then we couldn't stop it or reverse it. But that would be a great achievement, just to be able to understand what triggers it, and we can stay away from that. We could have a drug or an injection that would prevent it. It would be nice to have the ability to arrest the progression. Like many neurological diseases, Alzheimer's is progressive. More and more neurons die. And so the symptoms and cognitive decline gets worse and worse. And so maybe if we knew how to prevent it, that wouldn't allow us to arrest it. Once triggered, maybe it would cascade out of control. So we would need the ability to freeze it in place, to arrest it. And that would be a great accomplishment. Those could be the same thing. They could be different things. But imagine if somebody first showed the early and mild signs of Alzheimer's disease, if we could give them a pill and stop it from getting worse, that would be a great achievement. And then finally, in the way that many of us think of as a real cure for a disease, is that we could take people with Alzheimer's and reverse and fix what they have, that we could restore their declined cognitive function and turn them back into who they were before. And so this is what I mean by here we are being Einstein now, imagining the possibilities. And what I want to do is walk you through some really quite basic first principles of neuroscience to think about how we should be viewing this and how we could narrow things down. All right? So one of the questions that always happens at times like this, and so I thought I would just get it out of the way in the beginning, is what's the difference between dementia and Alzheimer's disease? So somebody was going to ask this, so here we go. So dementia is a blanket term meaning cognitive decline because of pathology in the brain, neurons dying. Alzheimer's disease is one kind of dementia. There are many other kinds of dementia. So everything listed here is a neurological problem that would rob people of their memory or cognition or their ability to control themselves, and it would be a form of dementia. And so Alzheimer's is just one. It happens, unfortunately, to be the most common, and the one where people most hope to prevent or cure. But that's the difference. Alzheimer's disease is one of many forms of dementia. Right? And so, of course, we're going to talk about Alzheimer's disease, but anything we talk about could apply to these other things. It could apply, I happen to care a lot about Parkinson's disease, another disease that involves neurons dying. And so we'll focus on Alzheimer's disease, but we could be thinking about any of the neurological diseases where neurons are dying. Now, full disclosure, I don't study Alzheimer's disease. It's not my area of expertise. But I do have a little skin in the game, and so I just want to tell you about that. So this is my grandmother, my late grandmother, Dorothy. She had Alzheimer's disease, and we watched her decline in her later years. This is my dad. He passed away 6 years ago. This was right about the time when I first started to notice early signs of Alzheimer's disease. In fact, I kind of sounded the alarm to my family, and unfortunately, I was right. This was about, 17 years ago, since my 17-year-old daughter was just born. That's my mom, Carol. She's still alive. You'll notice that she's holding her hand. As it turns out, she was just a few years away from her diagnosis of Parkinson's disease, and she had a tremor, and she had gotten into the habit of kind of holding on to her hand because it was starting to wiggle a little bit. Right? I'll skip my usual. That's my brother. I'll skip my usual joke about we don't know what's wrong with him, but it's a good one. So I care a lot about Alzheimer's. I care a lot about Parkinson's disease. I'm not an expert, but part of the passion for me is that I've been a neuroscientist for a long time, and in the beginning it was just a great way. It was just a fun thing to do, a great career to have. And as you can see, somewhere along the way, it got a little personal, and so we're all in this together, unfortunately. So let's drill down on Parkinson's disease. There's a lot of possibilities, a lot of theories, but I'm going to run you briefly through the best accepted set of ideas about what might be causing Alzheimer's disease, and they involve two proteins in the brain, beta amyloid and tau protein. And there's a couple of reasons why these are such strong candidates. One is that there are genetic forms of Alzheimer's disease. The early onset forms of Alzheimer's disease are genetically produced. And the change in the genes in each case, one way or another, relate to beta amyloid and tau protein. Number one. Number two, if we look at the brains of people who have died from Alzheimer's disease, we see two characteristic problems. One is a whole bunch of dead neurons, but we see these plaques, amyloid plaques, which are made up of a whole bunch of beta amyloid proteins glommed together. So this is what a plaque in the brain might look like, and it's just made up of beta amyloid. And then tau protein tends to make these neurofibrillary tangles. Neurons die, and they're surrounded by these kind of stripy layers of tau protein. Now, in the dish experiments, we know that beta amyloid and tau are both fairly toxic to neurons. And so it's sort of easy to imagine that that's the main problem, is that why are they there, and that's why they're killing neurons. But there are a lot of other possibilities. And I've just list a bunch here. There's theories about inflammation and infection, herpes. There's some evidence recently that infections such as herpes and even gingivitis can elicit inflammation reactions in the brain. There's a whole set of theories about iron accumulation in neurons and hyperexcitability. There's on and on and on. There's some about sleep being important for clearing beta amyloid, and people that have sleep problems should be concerned. Who's to say? Here's what I know. My dad had high blood pressure, he had gingivitis, and he had sleep apnea. So, I'm not unconvinced by any of these possibilities. There's also this interesting hint with sex differences. So women are about twice as likely to get Alzheimer's disease as men. Don't know why. But when men get Alzheimer's disease, it usually progresses more severely and rapidly. And so there are some hints there. So if we focus on this main idea that most scientists have pursued for a long time, the beta amyloid thing, even there, we can't seem to agree on what might be going on. So beta amyloid and tau are toxic to neurons. We know that. There's some evidence that tau protein helps make beta amyloid more toxic. So maybe there's some way that they team up together to cause problems. There's evidence that beta amyloid and tau kill synapses, and that killing those synapses makes abnormal activity in the brain. And maybe it's that abnormal activity that cascades out of control and causes neurons to die. There are theories about how beta amyloid spreads like a prion through the brain, like mad cow disease, and kills neurons in sort of this weird way of spreading from neuron to neuron, the so-called amyloid cascade hypothesis. So we can't even agree on what's going on with amyloid and tau. But let me show you sort of the textbook way that people think about things, and then tell you a little bit of bad news about that. Okay? So again, we're going to focus on beta amyloid and its tendency to form these plaques, and tau to form these tangles. And here's a little bit of what's known about beta amyloid. So, like many things in cell biology, it begins with normal processes. So there's a protein that's important for neuron function called amyloid precursor protein, APP. And like many proteins in the brain, it does some things itself that are important, and it also can be cut up into pieces, and then those pieces can be important. And APP is an example of that. And there's a class of molecules called enzymes that always end in tase. Beta secretase, alpha secretase, gamma secretase in this case. And these molecules they will cut proteins in particular places. And so this is a cartoon of an APP protein, and how beta secretase cuts it right here, and alpha here, gamma here. Now, as it turns out, in normal function, when things are going well, alpha secretase kind of dominates, and so it's often APP is cleaved into two to make these two molecules with their names. And then gamma secretase comes along and cuts it in its spot, and what we end up with is we have some APP molecules, we have some of these, and we have some P3 peptides that are floating around because of the way things have been cut on the outside of the neurons, and we have this AICD molecules floating around on the inside. Now, none of us, maybe only a couple of us here possibly, are molecular biologists, so we're not going to dive into all of this, but this is normal stuff that's happening. Here's the cartoon of a neuron membrane, and some of these are embedded in the membrane, some of these are floating on the outside, some on the inside, and there's a lot known about how these different molecules help make things happen for neurons. But then sometimes, for unknown reasons, things get a lot out of whack, and rather than alpha secretase dominating, beta secretase dominates, and there's too much cleaving at this spot, which instead of here. And so now you get these different molecules, and then gamma secretase comes along and does its normal thing. So instead of having these little P3 fragments like this as the final product, we end up with these bigger pieces, and this is the beta amyloid peptides. And that's bad because they have a tendency, again, for unknown reasons, something can trigger them into starting to clump together, and as they clump together, they become toxic to neurons. And as they clump together, they form the plaques that are such a prominent part of what we see in the brains of Alzheimer's patients. All right? Okay, well, that looks straightforward. You may be imagining what scientists imagine then is that, well, time to figure out a way to stop this beta secretase from, right? Didn't that make sense? Shut that down, and life's good. Alzheimer's cured. So here's a quote from a recent review article that I found, and BACE1, this is one of the categories of this beta secretase inhibitors, and we can just read it together. A quote from the end of this paper. Brain penetrant base 1, so ones that can get into the brain, inhibitors have been developed and clinical trials are underway. Both safety and efficacy are questionable. Several clinical trials suggest that BACE inhibition and other immunotherapies to reduce brain beta amyloid are insufficient to improve cognition in Alzheimer's disease. This may be due to the emphasis on the amyloid hypothesis despite big failures. That's a little dig to a whole field. There's been, in the last 10 years, just this whole horrible cascade of failures with trying to shut down beta amyloid as a way to prevent Alzheimer's disease. We may have to seriously consider shifting attention to therapeutic strategies other than beta secretase inhibition or reduction of beta amyloid alone. So this is the problem. This makes so much sense, and it's there. Early onset genetic forms of Alzheimer's disease clearly have these as one of the key pathologies and the defect in the genes. And yet attempts to prevent this from beta amyloid from forming, drugs that help clear beta amyloid from the brain, none of them are working that well. They don't seem, and we don't know, so we don't know. They don't know. Maybe the hypothesis is wrong. Maybe it's right, but there's other pieces. Maybe it's right and they're just not starting early enough, but here we are. A whole string of disappointments over the past 10 years from this whole beta amyloid hypothesis. So, of course, now when we go back to the list of things that we were thinking about then, we might put beta amyloid and tau in smaller letters, and that might help us start thinking about these other things, and maybe we should. But here's the problem. So what do we do? What do we do? So very few of you here are neuroscientists, and so you're just watching this as a hopefully at the end of the day slightly better informed bystander, but you're just watching this and waiting for these scientists to figure this out. Even the neuroscientists among us, we're all sort of wondering, what's going on here? How do we make sense of this? And one of the things about this, one of the things that makes us vulnerable in situations like this, is that people can swoop in and make claims. Right? If we don't know, if the main scientists, the big name scientists who are studying Alzheimer's disease are hitting dead ends, well, then that leaves room for other people to swoop in and start making claims. And we want to encourage anything that might help on the one hand, but we don't want to be fooled by shysters on the other hand. So what do we do? So what we do is we inform ourselves, and we do this thing that I talked about in the beginning. We're going to be like Einstein, and we're going to get good at using our imaginations about what might be possible and what's not, because that will help us evaluate what we see and know who might be up to something encouraging and who might be trying to pull the wool over our eyes. So that's the idea. And I thought we'd focus on memory, because memory is a key part of Alzheimer's disease. It's not the only part. As it progresses, people not only lose their memories, but they lose their ability to function cognitively and even to be aware of what's going on. But memory is so important and so fundamental to us. And if you think about, we could read these things, but really what I want to say with this is that we take it for granted how important memories are to us. But if you think about it for a second, if somebody scrambled your memories, you just wouldn't be you anymore. It would disconnect you from your past, from your family, from who you are. You wouldn't know what to expect next. You couldn't think about the future. You couldn't imagine what might be coming next. You just wouldn't be you. I really don't think there's anything more fundamental to our feeling of self-identity than our memories. It's our archaeological dig of our lives and who we are. And so things that rob our memories are horrible, and how our memories work are really interesting. And so that's what we're going to dive into. So different kinds of memories are stored in different ways, and that's been a topic of previous brainstorms. It could be a topic of one in the future. Motor memories are stored in the cerebellum. Other memories in different places. Fear memories may be in the amygdala. But the kind of memories that we think about, your memory of how you found out about this brainstorms, for example, the kind of cognitive memories that we have, those are largely about the cerebral cortex, and in particular this temporal lobe that sits on the side of the brain here, like behind, inside of your ears, and a structure buried deep within the temporal lobe called the hippocampus. And so let's dive down a little bit and think about that, and think about first just how neurons work, because we can't really think about the temporal lobe or the hippocampus or memory or anything neural without thinking just a little bit about how nerve cells work. So that's what your brain is. Your brain is the organ of neurons. You have about 80 billion neurons. They're little electrical cells. They generate electrical signals. And depending on how you count, you might, and my colleagues here may frown because they may think of a different number. But you have on the order of maybe 100 or 200 different kinds of neurons in your brain. 80 billion copies of about 200 different types. And by types, it means that they would be shaped differently. That this is a neuron in the hippocampus with the beautiful dendritic tree. This is a pyramidal neuron because its cell body is shaped like a pyramid. You can tell that it's a cortical neuron because it has these little teeny spines all up and down this tree-like dendritic tree. Different neurons would have different shapes. They're also different in the way that they wire with other neurons differently. Neurons talk to each other through synapses, so they generate electrical signals, and then those signals mediate communication to other neurons through synapses. So this is an artist's rendition of a synapse. It's the artist's rendition of one of these little spines blowing up. So here's the sending neuron that might send a signal down. These little green things here are the artist's version of the transmitter, the vesicles holding the transmitter that would get released. When it's released, it would talk to this neuron, and we'll talk about how it nudges its electricity to influence its signaling. But it's hard to underestimate the vastness of the brain and its processing. 80 billion neurons. You have on the order of about 500 trillion synapses in your brain. And learning and memory is mediated by neurons changing their strength. So, and we're going to talk about this. So, as you learn, as you experience, as you change as a person, those 500 trillion synapses, according to rules that are wired into them, change. And as they change, then that mediates your memories and the way you change with experience, and it's how you learn. And so you are the particular combination of the strengths of these 500 trillion synapses in your brain. And we're all share in common that our brains follow the same sort of rules. We all have the same kinds of neurons wired up approximately the same way. But we're all individuals because the wiring, each one of us has a slightly different variation on those themes of that wiring. And then, of course, we're all very different because we have a lifetime of experiences changing the strengths of these neurons to make us who we are and what we have as memories. So let's talk just a little bit about that signaling. This is my favorite cartoon that I got off the internet for that. So we're just imagining two neurons, and they generate signals called action potentials. I'll show you that in a second. And we're just imagining this little circuit in the brain where here's a neuron that is the so-called sending or presynaptic neuron, making a synapse on to the receiving or postsynaptic neuron. So when this neuron generates signals, it releases transmitter to nudge the electricity of this neuron, and it would use those nudges to think about whether it would make signals or not. So what do these signals look like? So this is a recording from an old friend of mine, Dave McCormick, and so we'll just let this run for a bit. What you're looking at, of course, as a scientist, I have to tell you what the axes are. This is voltage. So this, if we measure the voltage across the neuron's membrane, we would see it going up and down like this. This is time. And this is an unusual neuron. Never mind, it's a thalamic neuron. It's a very crazy neuron. But it does what neurons do, which is it makes these signals. And so you see each of these blips of voltage. These are the action potentials or spikes. What you can imagine then is neurons are thinking down here. They're thinking down here. They make decisions about sending signals when you see these blips. And you'll notice that when the voltage goes up, the neuron is more likely to send the signals. And this is important because it helps us understand the role of synapses. So then let's go back to the cartoon. And what I've drawn for you is some fake computer-generated signals. And I'm imagining the blue one here. This is one of those spikes. And we would record it in this presynaptic neuron here. And when it happens then, we would see that just after it happens, the black trace, if we were recording a signal in the postsynaptic receiving neuron, we would see this little nudge upwards. Now, that nudge upward is excitatory because it's nudging the voltage in a direction that might make this neuron generate signals. And now, what we mean by changing the strength of the synapse. So I'm imagining that we make that synapse stronger. What I would mean by that is that the next time an action potential came along and some transmitter released, the nudge would be bigger. That's all we mean by a stronger synapse, is that the nudge could be bigger. Why is that important? Well, the nudge could get big enough that just an action potential in this neuron would nudge big enough to make an action potential in the receiving neuron. Synapses can be inhibitory. So let's just pretend now this is an inhibitory synapse. So now here's the arrival of the action potential. And now the nudge is downward. And that's inhibitory. That's telling the receiving neuron, no, you don't generate a signal. It's encouraging it not to. And these synapses can get stronger and weaker too. I've drawn a couple here. Here's a weaker and here's a stronger inhibitory response. Right? And so this is what neurons do. They talk to each other. And in this cacophony of speaking, they get nudges, excitatory and inhibitory nudges, remembering now that a typical neuron might have 10,000 synapses onto it, 100,000 synapses onto it. All these nudges, upward nudges in saying be active, downward nudges saying don't be active. And all of those nudges can get stronger and weaker through learning and changes in synapses. So that's sort of the language of neurons. And so now what we can imagine, of course, I picked an example that's the exception. I should tell you now that these are nudges, but in fact, some neurons are designed to generate their own activity without any nudges. And in fact, most of this activity is spontaneous in this particular kind of neuron. So there can be nudges, excitatory nudges, inhibitory nudges. Neurons can have their own mechanisms that say, I just kind of like to be active spontaneously, and then the nudges can make that go up and down. So all of these different ways of doing these things then, all then in the context of neurons being wired with each other in these complicated ways, passing signals amongst one another, and that's what's going on in our brains. So, and we're going to get back to why that's important. This is going to be one of our key insights is thinking about that and why that's important. So now let's take a step back. So now this is an artist's rendition of a brain. There's 80 billion neurons inside of there. And now we're going to think about different functions. So there's motor functions and sensory functions, and here's vision back here, coordination. And a lot of conscious memories are about this temporal lobe here in the hippocampus underneath. And so let's take a step back now and think about what goes on there. And I want to start with the hippocampus, in part because it's a key part of memory, and in part because it's a key part of understanding the early symptoms of Alzheimer's disease. So the hippocampus is buried deep in this temporal lobe. It's not in this cartoon. If we were to cut a slice through here, then we might see something that looks like this. And this is one of these beautiful old hand-drawn renditions of the hippocampus. So there was a famous neuroanatomist, Santiago Ramon y Cajal, and he lived in an era where he had the ability to stain brain tissue so that only some of the neurons were stained. That's why you can see them as individuals. And he had a microscope, and he was a brilliant artist. And so he spent his days staining neurons. Here he stained the hippocampus, and he just drew the neurons. And he was the person who first proposed that brains are made of neurons, and there's different kinds of neurons with different shapes. So here's one of those pyramidal cells that I showed you earlier, right here. This is a hippocampal pyramidal cell. These neurons are smaller and more densely packed, and you can see from his drawing that they send their axons around this way. Well, these neurons make synapses onto these neurons. In fact, these neurons make synapses onto these neurons. And so what he saw was the brain is made of neurons. There are different kinds of neurons, and there's clearly a wiring diagram. And as it turns out, this wiring diagram is very important for learning and memory. I want to show you, this is the modern version of this. This is a mouse hippocampus in a mouse that was genetically altered so that some of these hippocampus neurons express a green fluorescent protein. So we're lazy now. We don't sit for hours and draw neurons. We just build a mouse where the neurons are green and we take a picture of it. But it's nice to know that 200 years have gone by and the hippocampus still looks like the hippocampus. So we know a lot about the hippocampus and memory from this guy. His name's Henry Malaison. We us neuroscientists all knew him as HM because when he was alive, his name couldn't be used in the publications. But the quick version of his story is that he had epilepsy as a kid, and to treat it, they removed his hippocampus on both sides, and which was not unusual. It had been done before on one side. He was the first person, as it turns out, to have his hippocampus removed on both sides. His epilepsy was severe because he had a trigger zone on both sides, both of his hippocampi. Well, that turned out to change his life quite a bit because from that day forward, from the time of his surgery forward, he never again formed another long-term memory. So it became immediately clear that this was true. A brilliant neuroscientist named Brenda Milner studied him for almost 30 years. And she showed a variety of things. She showed that his memories that he had made before his surgery were as good as yours and mine. He could remember being a kid. He could remember the bicycle accident that may very well have been why he developed epilepsy. So he could describe the neighborhood that he grew up in and draw maps of how he used to bicycle to places and such. But from the time that he had his surgery forward, never made a new memory. And so I saw Brenda Milner give a talk one time, and she told this anecdote. She'd been working with him for about more than 20 years. And she brought another colleague in, and that colleague knew the whole story, knew everything, but just didn't realize what it meant when you saw it. And Brenda Milner walked in, Henry stood up, and again, they'd been working together almost daily for 22 years, and she walked in and said, "Good morning, Mr. Malaison, I'm Dr. Milner, I'll be working with you today." And he said, "It's nice to meet you." Now it's interesting, and this was the brilliance of Brenda Milner. He reported things like, "You sure seem familiar to me." So there were memories there, and she showed a variety of ways that his memories were intact. What he didn't have are these conscious memories that we make so much use of. And one of my favorite examples of that is that she tested him on this game, the Tower of Hanoi game. Did anybody not know this? It's a real simple game. You've got these discs of different sizes and 3 poles. Every move that you make involves moving a disc from one pole to the one adjacent to it. And the task is to start with them here and end with them here, stacked up in the order. And the rule is that you can never put a bigger disc on top of a smaller disc. Now this is the 1, 2, 3. This is the 7-disc version. So I think that, what is 7? 2 to the 7. Thank you. So if you were UT undergraduates, you would have done it even faster. And with more glee. Turns out, so the ideal solution requires 128 moves in a particular sequence. So she brings him this one day and says, "Have you ever played this before?" No. He plays with it and isn't very good. She brings it back in day after day. Every day he reports he's never seen it before. You ever played this game before? Never even seen this game. But over the weeks of this experiment, he got better and better until she would bring it in. He would report that he had never seen it before, and then he would solve it in 128 moves. And probably say something like, "Well, I'm pretty good at this." But so it was Brenda Milner who showed us that there's different kinds of memory stored in different parts of our brain, mediated in different ways. But the hippocampus is important for the memories that we're most familiar with, our conscious ones, the ones that we can recall things and report to each other and imagine. And so we need our hippocampus not apparently to store memories, but it's not where they're stored, but we need them to store them. So then the sort of common idea about these kind of memories, and my neuroscientist friends know that I'm oversimplifying things. Here's our cartoon of the hippocampus, and here's one of Ramon Cajal's drawings of neurons in the cerebral cortex. Let's say that this is from the temporal lobe. Then the idea is that processing all the action potentials and synapses and signals bouncing around in the hippocampus are to store memories, or about storing, but the storage itself eventually takes place in the synapses in the cerebral cortex. All right? So hippocampus for storing, and then cerebral cortex being the place where they're stored. This is why for HM, with his hippocampus removed, he can't store anymore, but he can retrieve the memories that have already been stored from his past before his surgery. So how does this relate to Alzheimer's disease? Well, number one, hippocampus is one of the most sensitive, hippocampal neurons are among the most sensitive to the ravages of Alzheimer's disease. They tend to die early. And that's why some of the earliest symptoms that you might notice are memory issues and navigation issues. And one of our panelists, Laura Colgan, will help us understand that maybe a little better. I saw this in my dad. My dad turned into HM. He turned into Henry Malaison. His symptoms eventually got bad enough that we moved him to Austin, which I felt really guilty about. And we had strategically positioned where he was living so that I could drive by there on the way home. And out of guilt, I would call him and say, Dad, grab your coat. I'll swing by and pick you up on my way home from work. We'll go have a beer. So I would go by and he wasn't there, and I'd go knock on his door, and he'd open the door and he'd say, Mike, what a surprise, I wasn't expecting you. And so I soon learned, no point in calling. I would just show up, and whether I called or didn't, he would say, Mike, what a surprise, right? And so that's why among the earliest symptoms are these storing new memories. Now, my dad also displayed the classic symptoms that while this was happening, he was very good at recalling his old memories. Right? Now, as Alzheimer's disease progresses, then it starts to kill neurons in the cerebral cortex. Then we find that existing memories then start to go away too. And we don't know why, but there is this phenomenon. I saw this in my dad very clearly, that the newest memories disappear first. And sometimes the phrase is the fog marches backwards in time. So I knew all the different places that my dad had lived in his life. And when I was interacting, he'll forgive me for doing experiments on him, I'm sure. But I would interact with him and I would engage him in conversations about those periods in his life. And we moved him here from California. And over the course of the first year that he lived here in Austin, California went away to him. And it was Minnesota before that. You can hear the O in my Minnesota. It was Minnesota before that, and another half a year or so went by and Minnesota went away. And by the time he was in real bad shape and some days could recognize me and some days maybe not so much, he could still, on a good day, remember being in the Navy as a teenager and being on a minesweeper in the Korean War. We don't know why that happens, but the newest memories disappear first, and the oldest memories hang in there the longest, right? That's about the disease progressing, hippocampal neurons dying first, and then cortical neurons dying, and then going away like that. And so, again, because the hippocampus is about the process of storage, and cortex is about storing, as the hippocampus dies first, that was the era when there was no point in calling my dad because he wouldn't remember that I called anyway. And then as it progressed into the cerebral cortex, California went away, and Minnesota went away, and Kansas City went away, and all of his memories went away over time. So that's a little bit about neurons. That's a little bit about memory. And so now we're at the point in the afternoon where I want to bring all this together and see if we can't arm you with a better understanding about how to interpret and make use of information that you see. So this is the part where we're going to try to be like Einstein and use our imagination and first principles to narrow down the possibilities. All right? So I'm going to go back to this diagram and just remember, of course, that this is such a simplification. 80 billion of these and 500 trillion synapses, and neurons having often 10,000 or 100,000 synapses and all that activity and everything. But then as neurons die, then that's when the cognitive abilities go away. Hippocampus neurons die early, so you don't make new memories. Cortical neurons die later, so that's why memories, old memories start going away, etc. But here's one factoid. Here's one factoid that you need to know to do that Einstein thing and dramatically narrow down the list of possibilities of what might happen for a cure in Alzheimer's disease. Alzheimer's disease kills neurons, so the neurons die. That's bad. So whatever that neuron did goes away. Whatever those synapses encoded, whatever the strength of those particular synapses encoded, some memory, participating in some memory, that's gone too. But it's worse than that, because when neurons die, they shrivel up. And as they shrivel up then, any semblance or evidence that we would have about how they entered into the wiring diagram and who they connected to goes away. So we could look at a neuron, if we could image a dead neuron in an Alzheimer's patient, we could use some fancy optical laser technique to look at those neurons. We could see this shriveled up dead neuron. Well, that's bad, it's dead. But from what we can see, we could not reconstruct the many neurons that it made synapses onto. We lose also the information about the wiring diagram. We have no way of reconstructing or figuring out what the strength of all those synapses were. And so we lose this information that's devastating. It's not just that the neuron dies, but we don't know who it connected with before, and we don't know what the strengths of those synapses are. And I hope you know enough now to realize that that means is that you can't get back what the person had before. So even if some brilliant scientists invented a way of making neurons come back to life, they wouldn't do it because they wouldn't know who to wire to. So we could have taken my dad and squirted some chemical in his hippocampus and made hippocampus neurons, living hippocampus neurons, appear where the dead ones had been before. But it wouldn't have been my dad, because it wouldn't have been the wiring that it was before, and it wouldn't have been that beautiful pattern of stronger and weaker synapses for his memories. It would have just been a human, and maybe one quite different from my dad. And this is really one of the most important problems with Alzheimer's disease, is that when neurons die, we lose so much. And so now if we kind of map that onto this hierarchy that I proposed before, and if we imagine the one that we hoped for the most, which is the ability to reverse the cognitive decline in somebody who's already gone down the roadway of Alzheimer's disease. If that's our definition of a cure, the ability to take somebody and fix them once it started to do its job, there will never be a cure that way, ever. Maybe in 500 years we would have the ability to do that, but here's the thing. Long before that, we will have accomplished these things, which are much more doable. So there we go. We've been Einstein. We've narrowed down the possibilities. When you read somewhere somebody claiming to have a drug that reverses the cognitive decline of Alzheimer's disease, be suspicious. When you think about and hope about and pray for scientists to find ways to deal with Alzheimer's disease, hope and pray and vote and fund attempts to prevent them from happening in the first place. We need to focus our abilities on preventing the neurons from dying in the first place. Or if that's not good enough, to arrest progression and or finding ways to stop the progression once it starts. But once those neurons are dead, we're done. And you know, it was a painful thing as a neuroscientist to watch my dad going downhill and to know that if they found a cure tomorrow, it wouldn't help my dad. right? And so that's bad, but it's focusing, right? We should be focused on preventing the death of the neurons and stopping the progression, and really, frankly, de-emphasize trying to fix things once they're broken. And so there's all sorts of, so I just found these on the web. So if I Googled treatments for Alzheimer's disease, and these are all a bunch of drugs that have gone through clinical trials to help the people with cognitive decline for Alzheimer's disease. Okay, so but I have to tell you, this is a drug company view of dealing with Alzheimer's disease, right? It isn't gonna reverse what they have, right? You now know why. It might make them a little better in the beginning, but if it doesn't stop the neurons from dying, then it isn't going to stop the progression of the disease, and things are going to get worse. And I put these drugs down here because, first of all, none of these drugs act specifically on Alzheimer's disease. These drugs make us perform a little better on cognitive tasks, the rest of us. And frankly, most of them, their effects are smaller than the effects of these drugs. I mean, there's a reason why we're all addicted to caffeine, because there's a lot of good evidence that we perform a little better on all sorts of cognitive tasks when we have the right dose of caffeine. Now, partly because my dad was pretty far along before we moved him here, but I refused all of these drugs, partly because he was too far along, and partly because if he had a couple of cups of coffee, he was going to get just as much benefit as any one of these drugs would have done. All right? So it's fine. I mean, maybe we should have these drugs, but let's not fool ourselves that this is where the cure is. This isn't the magic. This is a Band-Aid over a really bad problem that's going to continue to get worse, right? I mean, imagine if I told you, it sounds silly, but how did you deal with your dad's cognitive decline and Alzheimer's? I bought him coffee, okay? That's as effective as any of these drugs. Sad truth. Now, I'm not here to sort of pile on any particular thing or person, but these things exist. And I took the name of this is a book that I found on Amazon, and I blanked out the author's name because I'm not here to pile on any one thing. But I haven't read this book. And I'm not going to read this book, and I know I don't need this book, because any book that claims that it's going to prevent or reverse cognitive decline, I know that this is somebody just making some money writing a book. And it makes me a little angry because it gives people false hope. And so this is part of what I want for you now, is that you won't be fooled by these things, and you should be thinking about real hope and not being fooled by these false hope, all right? I'm sure whoever wrote this book made millions writing this book, and probably got donations from desperate people all over the country, thinking that, well, here's somebody onto it. Those scientists aren't doing it. And that's fine, but it isn't going to help. All right, we should be focusing on things that stop neurons from dying. So, like I said, we'll wrap up here. Like I said, we've focused on Alzheimer's disease, but if you care about other dementias, or Parkinson's disease, or ALS, or any number of those diseases where neurons are dying, it's the same thing. When I think about cures for Parkinson's disease to help my mom, I think about preventing the dopamine neurons from dying. Those are the neurons that are dying. Don't know that there's anything that's going to reverse my mom's symptoms, but if they could find a treatment that arrested the death of the dopamine neurons tomorrow, my mom could take it and not get any worse. That's what we have. And so we should always be focused on prevention and maybe on arresting progression. And we should be fooled, I think, less by claims about reversal or cures or fixing. Okay? Whether it's Parkinson's disease or Alzheimer's or ALS or any of these things, we have to stop the neurons from dying. So, if those of you who have been in Brainstorms before, what we're going to do is we're going to take just a couple-minute break. I hope at least some of you grabbed a 3x5 card on the way in. The idea is that if you have a question you want to write down, if you want a 3x5 card to write a question, what we're going to do is the panelists are going to come up and I'm going to introduce them. Our volunteers are going to run around and grab the cards. And what we're going to do then is you're welcome to come to the microphones and ask questions. The panelists are going to be looking through the questions and grabbing, and we're going to just do Q&A until they kick us out of here. Okay? And so with that, thank you for your attention, and we'll start. Where did our volunteers go? Volunteers! Anybody not have a card but would like to write down a question? If you're willing to come to the microphone, that's the alternative. But we do the cards in part because we like to read your questions, and in part because some people don't really want to come to the microphone. So let's take a couple of minutes. If you've done a question, pass it to the aisles. If you want to write one down, do it really fast. But then I think what we'll do is, while we're doing that, let me introduce our panelists that I've invited to help with the questions today, and then we can get started. So one of our panelists is Dr. Laura Colgan. Laura is an associate professor in the Department of Neuroscience, and she's in the Center for Learning and Memory here at UT. And she's one of the world's authorities on the hippocampus. And so she's going to obviously be quite valuable for your questions. If you have questions about the hippocampus and plasticity and brain rhythms in the hippocampus, which is our other big thing, she'll be a big help with there. Second panelist is Dr. Nace Golding. He's a professor in the Department of Neuroscience, also Center for Learning and Memory. Nace is a neurophysiologist. He makes those kinds of recordings that you saw in that movie that I showed. And he does those recordings in the context of trying to understand how we localize sounds and how we learn to localize sounds. So he and I are a little further away from hippocampus and Alzheimer's disease, but between the three of us, we're going to take a good stab at answering your questions. All right. So volunteers, if you can keep gathering, we'll just kind of do this dynamically. Why don't we start with some questions from the microphone, and then you guys don't have any cards yet? Okay. So anyway, go ahead. Oh, you're for a card. Okay, so why don't you start us off, please?
Hi there. I'm a speech pathologist. I work with adults with brain injury. So I was a little confused about the implication of a damaged brain cell, meaning that the person is just irreversibly not them anymore. I had a little trouble processing that because I work with people with damaged brain cells, and they're still them. Their brain works differently, and they learn differently, but they are still them, and they regain skills, as well as cognitive and motor skills through our work with them. They learn strategies. They also regain skills. They make new pathways in their brain. So I wonder if you could kind of speak to that. I am in the process of reading The End of Alzheimer's as well, and I think it's worth the read. He is a neuroscientist, and he has a lot of really compelling. He's very. I don't think you would disagree with it quite as much as you think you might. He speaks very intelligently to it, and I don't think he's quite selling anything in the way that it's. Well, but it's a very compelling book.
I'm sure it is. Let me do so. Different parts of the brain do different things. When we lose neurons, we lose that part. That's not to say that we can't do alternative strategies. So I study the cerebellum, motor coordination. Over time, people with cerebellar damage learn to be more coordinated again, but never in the way they were before. They learn an alternative strategy. Okay? It's one of the reasons why I focused on memories, is because memories, they're gone, right? So that there's no alternative strategy to gain back a memory that you had before. So part of your question is about different parts of the brain doing different things. And when we lose something, we can learn an alternative strategy to get a semblance of that back.
So isn't it possible that maybe is what's happening with the reversal of cognitive decline, potentially, that addressing the inflammation or whatever the process is. He doesn't believe fully in the beta protein thing, so it's possible that perhaps alternative strategies are forming in the brain to prevent presentation the cognitive decline.
If he has a way to think about how to stop neurons from dying, then we should listen to him.
He does, yeah. Okay. It's worth the read.
Yeah, Mike, thank you for the talk. My question is it's going to be a little different. It's really about computational modeling. So there's been some publications recently from the ophthalmology literature on retinal biomarkers, which may help with early diagnosis of Alzheimer's disease. And I think about the visual pathway. It's also a system which has been modeled and studied extensively from a computational modeling viewpoint, right from retina to the cortex. So given these potentially promising developments in diagnosis techniques, detection techniques, could there come a time when the computational models we have could be enhanced, augmented, so that we could actually predict, using these kinds of high-resolution retinal imaging, the progression of the disease, maybe even leading to better or focused treatments for Alzheimer's disease?
Yeah, I think that's a really promising avenue, and certainly there are people that are working on that, as you mentioned. If there's biomarkers that can be put into computational models that can then predict the progression of the disease, it sounds realistic to me. Yeah, I think it's a promising research avenue. That's a good question. Was there a beginning part of your question? I'm sorry if I missed something.
No, the beginning really was that could we it's basically, I think you just answered, could we computationally be able to better catch, the disease early, know what it may do more precisely, and then, maybe tailor treatments to it, right?
Yes, so I think that's a promising direction of research, definitely.
So I think one of the ways that could come into play is the ideas for Alzheimer's that are about early damage leading to abnormal activity, leading to a lot more damage. Those kind of models would be really good for that, because if you knew how to damage a model of the brain in that way and you watch that activity cascade out of control, that could be very informative. So.
Thank you.
I noticed in your slide, one of the possible causes was aluminum pots and pans, which of course we've been told not to use now for about 50 years, even some arthritis potential from that. So my question is there any ongoing research about other toxic metals that can cross the blood-brain barrier, such as mercury and thimerosal and aluminum in vaccines and flu shots?
The most interesting talk that I've seen in the last couple of years was by an Australian researcher who is an advocate of the idea that iron accumulation in neurons can lead to a lot of these diseases that involve death of neurons. The list that I put up there, that was everything that I could find on the internet. Some of those have better scientific underpinnings than others. I think the aluminum one was more or less debunked in the 80s. The mercury one, I think, was about the same time. And so I think your typical Alzheimer's researcher would kind of shrug and say, we kind of eliminated those.
Thank you.
Thank you for the series of talks. This is my third one.
Great.
I found them really fascinating. I'm here today specifically for a very personal reason, because my 74-year-old father was recently diagnosed with Alzheimer's. Not a surprise to us, because we've been noticing the cognitive decline for the last 2 or 3 years. And this is a college professor, a very intelligent person, et cetera. So I have 2 questions. One of them might not be answerable, so feel free to say so. And that is, through your all's research and I'm assuming your interactions with people or knowledge of people who are experienced in Alzheimer's, do we have a sense of what the experience is for the person who actually has Alzheimer's? And you're nodding your head, so I'm thinking maybe it is answerable, so I'll skip my second question.
No, I'm nodding my head because I think it's just an amazing question. And
Well,
as I watched my dad go downhill, I couldn't stop. I mean, I couldn't stop thinking about that question. I think it's a brilliant question. You guys have any ideas?
I would say that I think it changed. I mean, so when I first graduated from college, I actually worked with Alzheimer's disease patients doing a study of ideomotor apraxia. And then for a year after that, I volunteered for an organization called Citizens for Quality Nursing Home Care. So I met a large variety of different Alzheimer's patients at different stages of the disease, and I think it's a really interesting question because you find some people that they can definitely tell something's going wrong because they're very, very agitated, and their family members would say that's not their normal personality, but then other ones that were just completely happy, and some that were even childlike, that maybe they had lost all their memories except their childhood memories, and they were just sort of, seemed extremely happy. And so, but I think we don't really know, but I think that they can. I feel like Alzheimer's patients can express their feelings to you. Sometimes they may not remember you, unfortunately.
So, yeah. I can't tell you how long I thought about the answers to that question. All I can tell you is that I found myself thinking out of the box. I hate that phrase, but the whole thing with not bothering to call when I came over, that's a funny one. But just things like this. So my dad always liked to read, and then I noticed that he was always, I saw him reading a lot, but he was always on the first chapter of whatever book he was reading. And the reason why is he didn't remember reading it, so he was always starting it over. So I thought for a while, and I bought one of my favorite books for him to read. And the one that I picked was one where the beginning is just really amazing. It's one of those books that just grabs you in the first 2 pages. And I figured if he was going to read the same 2 pages over and over and over again, they might as well be really killer, 2 pages. Right? And he did comment that he really thought that was a great book, but then couldn't report to me, of course, why it was great or anything like that. So, I don't know the answer to your question, but I'd say be creative. Think of ways that you can tap in as their world gets smaller. Think of ways of making that smaller world nicer. That's the best that we could think of.
Thank you.
You guys have a card question that looks cool? No? He's going to turn it on for you.
Here we go. So one question is, how valid are commercial genetic tests like 23andMe? Is there a benefit from knowing many years in advance? And I think there is a genetic basis for at least some forms of Alzheimer's, but others not. And so a test could actually reveal, although I think nowadays they actually will not tell you whether you have...
Not unless you check the box. Yeah.
Unless you check the box, whether you actually have the genetic mutation that would actually predispose you to the genetic form of Alzheimer's disease, genetically inheritable form. Is there a benefit to knowing many years in advance? Of course. You would plan your life very differently based on that knowledge. But I think probably most forms of Alzheimer's, you really won't know from a genetic test when or to what degree you're going to actually, or if at all, that you're going to suffer from this disease.
Yeah, it certainly relates to the previous question about ways to detect early onset. Until we have ways of stopping the progression, detecting early onset is less powerful. If we had ways like that, then we would focus, early detection would be incredibly valuable, right? So,
I have 2 questions. One, are there any reasons why aluminum pots and pans and aluminum and deodorants should be avoided because of Alzheimer's or multiple sclerosis. And I know you touched on that, but it was so different from what I've read, so.
I mean, can I step in just for a second? I'd like to preface, because I'm reading a lot of these questions. We're not medical doctors. We're PhDs who are doing basic research. So and we're also not environmental scientists. So I would say sometimes we might feel a little bit uncomfortable answering some of these questions. Like clinical things, I'll tell you up front, I'm not an MD, so I don't feel comfortable answering those questions. I think it would be irresponsible of me to do so. I can talk to you about research that I know about and I'm an expert about, or I can tell you some anecdotal things like I did about the studies with Alzheimer's patients and tell you specifically what my experience was to where I got that knowledge. But as far as Alzheimer's and aluminum in deodorant, I'll tell you, I wear regular antiperspirant for what that's worth. But I think that if there's robust research about that, I have to be completely honest with you. I'm not an expert about that, so I don't feel that my comment about it would be helpful to you. I'm sorry.
I agree. Let me share with you a little anecdote. So I was having lunch recently with one of our Department of Neuroscience donors, and smart person, asked her what I thought was a really clever question. His dad has Alzheimer's disease. That's part of why we were having lunch and talking about things. And we talked about things like, what can I do to help my dad? And we did that part of the discussion. Then he looked at me and said, he said, I know you're not a doctor. I know you don't study Alzheimer's disease, but as a neuroscience, what do you do? What do you do to protect yourself? Right? So that's the best we can do is translate what we think we know into how we deal with our own lives. That I'm comfortable with talking about. Sleep, keep your blood pressure down, stay healthy, exercise. Those are things that all have proven beneficial, protective properties. All right? I don't know if our pans are aluminum or not. I don't know if my antiperspirant has aluminum in it or not. But those are the things that I do, because I know the data that says that it really matters that you do those things.
My second question is, what is late onset Alzheimer's?
So early onset Alzheimer's is defined usually before the mid-60s, and almost all forms of that are genetic. They have a genetic predisposition. Late onset is regular, idiopathic, meaning of unknown origin Alzheimer's, and those are the ones that start when we're older, and the incidence grows rapidly as we age. So people in their 90s have a much higher chance of developing Alzheimer's than people in their 70s. But that's what it means. It's the one that start late and don't have an obvious genetic component.
And yet 23andMe is sending out things saying you've got one gene to show that the higher probability of late onset Alzheimer's.
Well, those are usually about genes, about APP, or the secretases and stuff, and they're going on suggestive evidence. There's some paper somewhere that said this maybe, and so we can say, well, maybe then that's the idea. But as scientists, we like things to be solid.
I think there can be an analogy with breast cancer, right? Like you may have the gene that you're at a high probability of developing breast cancer, and some people may decide then to do a preventative mastectomy, but you may have that gene and you never get breast cancer. So I think that would be a good analogy.
Ma'am?
Is there any statistical research connecting dementia or Alzheimer's prevalence to certain industries or chemicals or inhalants, particularly inhalants? Because when I sold private health insurance 20 years ago, I was quite surprised to find that the highest rates for personal insurance were for beauty salon people and motorcycle riders, but that was But I didn't expect to find the beauty operators in the same category with the motorcycle riders. And inhalants, particularly, since they go straight to the brain, I just wondered if anybody had researched that particular connection or possibility.
I'm not as we say, we're not experts in this particular area, but I'd say that these kinds of connections are extremely difficult to make because these are chronic use issues. People have other things that they're associated with. They have long lives, and so actually, by the time Alzheimer's manifests itself, it's extremely hard to make the connection, to go backwards in time and know exactly what it was about their lives that actually led to, or whether there was any connection with their predisposition for Alzheimer's or not. So these kinds of connections are extremely difficult to make, and they typically require massive studies involving many, many patients so that they can weed out all the other variables of the idiosyncrasies of the way people live their lives and what they're exposed to. And that's not a very good answer, not a complete answer, but what you're asking is a very difficult question, a very difficult question to answer from a medical standpoint.
And I think some of the things with insurance rates, it might be more because they've been linked to cancer, because I know we use a lot of chemicals in the lab that you could inhale the fumes, and we have material safety data sheets for all those chemicals, and they're all like possible carcinogen, or like heavy metals that we use in the lab to plate our recording devices, possible carcinogen. So some of those things that I think there's more things that have been linked to cancer correlationally than to Alzheimer's disease, to be honest with you, for the reason that Nace has mentioned.
Okay, thank you.
Dr. Mauk referred to the lack of success with the drug companies exploring the amyloid aspect of Alzheimer's prevention or cure. A recent article in that scientific journal called Fortune magazine talked about there's a new group of researchers out there being funded by the usual cast of Silicon Valley billionaires who are exploring prevention protocol that involves L-lysine. I thought that was interesting. They made a reference to, Amazon, you can get it for $50 for 100 pounds or whatever. Have you heard about that at all, or anything you know of? They also are linking the prevalence of it to exposure to blue-green algae, which is everywhere. It's when you go swimming. So just a question. Again, it was one of those 20-page articles that was kind of fascinating.
Yeah, I read that article.
Did you?
Yeah. The trouble is there's thousands of articles like that. So what do we do? What do we do with all that, right? So one of them may be right, but how do we process that? That's the frustration for me, is we could sit down and find 50 articles in the last 5 years about things like that. And many of them are attracting lots of funding to study, right? Even as a neuroscientist, I have no idea which ones to be excited about. It's the hard part.
Yeah.
Something I did want to mention, as far as the experience of the person who has Alzheimer's, my aunt has Lewy body dementia, and her husband has been very involved in that area, not scientifically, but personally, and participated in a study through the University of Washington that through different sort of virtual reality processes help people kind of understand what a person who is losing some of their experiential information, losing some of their memory, goes through. And I thought that was a very noble thing for them to pursue, because somebody who has dementia, be that Lewy body, be that Alzheimer's, be that frontal temporal lobe, it's pretty devastating. My mother has not yet been diagnosed with Alzheimer's, but has been diagnosed with mild cognitive impairment, and that was done through some neuropsychological testing. And it's been very interesting watching how she navigates. My sisters and I have found a website and app that helps us communicate with each other, and we've taken some of the, as the geriatric psychiatry nurse practitioner recommended, that she give away some of the things that make her annoyed. And so we've taken that on. What I find so interesting, and I actually wanted to ask Dr. Colgan about, was what I've observed from her, and just FYI, I'm a nurse practitioner, and so observation is what I do. I've noticed that while her immediate memory formation is gone, and that was verified in the neuropsych testing, she is able to make memory through various means, which I find really interesting. We'll write things down for her. She's got things on a calendar. So repetition of the information helps her store some of the things that we're talking about. And so she is able to refer to some aspects. I haven't got all the details down yet. I wonder what you think might be happening there.
So I'm actually going to take the opportunity to link your question to one of the cards because they go together quite nicely, which you're saying that although she's unable to form new memories, you feel that she can have strategies involving extra repetition or extra rehearsal, or or not relying on the memory by writing things down, of course, is another strategy. But I think that's linked to this question that we have on the card. Intuitively, why do you think older memories last longer? So one idea about memory that has quite a lot of experimental evidence is that memories are actually, every time you retrieve them, you reactivate this memory in its storage place, and it can actually be sort of consolidated again and linked to other similar memories. So it gets more and more distributed throughout more and more sites. So I think that it could be one explanation why, sort of every time there's a rehearsal, if there is anything there, it's going to get retrieved because it's going to be recognized as similar to what was seen before or what was, rehearsed before. And similarly with older memories, the older our memories are, the more that we've thought about them over and over again. You're remembering your birthday party from your childhood, fun birthday party or whatever. There's tons of other stuff from our childhood that we never remember, but we don't know because we don't remember it. Like we don't know what we've forgotten. But I think that the older memories are, the more likely they are to have been distributed throughout all these different brain sites and linked to other similar memories. So they become kind of more robust to being forgotten. They also become vulnerable actually to being less reliable because every time they get reactivated, they can be sort of monkeyed with, interfered with. But nonetheless, you usually don't know that happened. So from your perspective, you would think you're still remembering it perfectly. But I think that there could be a real biological reason why those strategies are helping her.
Thank you.
All right, those of you who have been to Brainstorms before, you know that it's time for lightning round. We've got a lot of questions and we're running out of time. And so we're going to challenge the faculty, ask your question. But the challenge for the panelists is you've got 20 seconds. I'll time. Thanks. Laura's so competitive. Who's timing? So anyway, you ask your question the way you want to, but we're going to find a way to answer it in 20 seconds. So
well, first off, thank you for your talk. I'm a physician, and I trained during the epidemic of AIDS, and AIDS and Alzheimer's carry a lot of similarities in the fact that they are fatal illnesses. We don't understand them. We didn't understand it then. And it subsequently results in people, I think, desperately seeking answers. And I appreciate your skepticism in your presentation because I think it's extremely valid. We read books that validate our belief systems and our hope that things will be discovered. So I do have a question, and this is parsing out for our PhDs here. If a patient is referred to your Institute for Neuroscience, how do you parse out the patient who has organic brain syndrome, multi-infarct dementia, and Alzheimer's clinically to then proceed with a treatment plan when frequently it's protean in its manifestation?
TikTok.
Well, so as researchers, we generally don't have patients. We're not a
We have
hospital.
brains.
Our patients are generally, rodent models and rodents that can't talk to us. So, from the clinical standpoint, I don't really know how to answer the question because this is not part of our experience as researchers studying very specific questions in rodent models. So, I'm not sure how to answer that question.
Well, then it makes it very difficult as a clinician to sort of say, let's throw a medicine at someone and say maybe, because it is typically protean in its clinical presentation. So I didn't understand. I thought y'all did evaluate patients, or at least referred to, say, from the medical school or something.
No, we're not in the medical school. We're in College of Natural Sciences, and we don't have a medical degree. As Nate said, we study mechanisms of, let's say, cell death or abnormal physiology in rodent models.
Well, thank you then.
Please.
Yes, given that Alzheimer's is strikingly more common in women, and that menopause occurs earlier in the life cycle than andropause, what do you think is the role of estrogen in keeping brain cells alive?
I don't know.
It's actually a topic that we've considered. So one of the things we want to start next year for Brainstorms is we may start inviting some neuroscientists from across the country so that we can cherry pick fun topics. And hormones in the brain is an obvious fun topic. So
Yeah, I'm a retired OB-GYN, so I'd be happy to help you get that
yay,
started.
okay.
Yeah,
Thank
it is actually significant that hippocampal neurons in these regions that are important for learning and memory are really richly endowed with estrogen receptors.
Yes.
And that actually affects processes of learning and memory. So it's really probably not an accident that there are sex differences.
And there is
these
evidence
problems.
that estrogen supplementation reduces incidence of Alzheimer's and dementia in general.
Yeah.
My name's Melissa, and I'm a speech pathologist, and I work with patients with Parkinson's. And I was just curious what research or how much research has been done regarding the gut-brain connection, or if there is any in Alzheimer's disease.
I mean, there's actually, again, I don't work on this specifically, but I know there's this guy at Baylor, and I'm blanking on his last name right now. His first name is Morrow, who's doing actually really excellent research on the link between the gut and the brain. But I think that the way that I like to think about it just personally, again, is me as a person, so take this as a grain of salt. I mean, our whole body, everything is connected. So what's bad for your gut is going to be bad for your brain. What's good for your gut is also good for your brain.
Thank you.
I would jump in and say that there are a lot of questions that actually had to do with what's good, what practices are good for our brains, or how can we prevent onset of Alzheimer's based on our particular patterns of exercise or diet or what have you. And as Mike said in his presentation, the brain is an organ, and it's really in many ways no different than all the other organs in our body. It has the same maintenance requirements, requires glucose and all the things that are used to maintain cells. So a lot of the same strategies that are going to be good for all of our organs are going to be good for our brain as well.
Including sleep, it turns out. Yeah.
It's funny because my question happens to be about sleep, so thanks for the segue. My mother suffered from sleep apnea and also blood pressure, high blood pressure since her 20s. She developed later onset Alzheimer's and dementia. I've been reading Matthew Weiler's, I believe I've got it right, Matthew Weiler Walker's book, The Power of Sleep or Why We Sleep. I'm forgetting the name. But I've become really fascinated with deep REM sleep cycles and the connection to maybe regenerating or helping our neurons in the brain. Can you speak to the necessity of having deep REM sleep versus the two types of sleep necessary, through several cycles to have a healthy brain? Do you know anything about that?
I can actually comment a little bit on this. I can't differentiate between the different types of sleep, but as Mike mentioned in his talk, when we're awake, we actually produce about twice as much tau protein as we do when we are sleeping. And it's thought that when we sleep, there are processes that help degrade that protein. And so for sleep disorders, or those of us who like to pull all-nighters, not me anymore, fortunately, you can imagine how this would actually affect the buildup of tau protein, because you're now compromising the processes that would actually break it down, and now increasing the duration of time that you're actually producing a higher level of tau protein, thus leading to more accumulation.
I just add one more thing there about the different stages of sleep. So during REM sleep, there are actually vascular surges that could potentially be sort of, flushing things out. And probably our 20 seconds is up.
Fourth straight brainstorms where we've proven that PhDs failed the lightning round test, but because the questions are, so we're going to have to make your question the last one. I'm sorry. So,
all right. My grandma recently passed from a long fight with Alzheimer's disease after 12 years. I was surprised to see how long it took. And she was taken to a center, to sort of a nursing home. She wasn't put there permanently, but she would go there 3 times a week. And they would do exercises to, keep her brain active. And I was wondering how effective brain stimulation in that regard. I think some of it was math operations, word games. I'm not sure what it was, but it was some sort of stimulation. And I was wondering how effective that is pre-onset, if, for example, you were to know that you're prone to that, and after the disease has appeared.
I just really hope that crossword puzzles matter, because then
it's probably not the case, but
I'm going to be good for a long time. But there's a lot of evidence that part of our normal cognitive decline is that our worlds get smaller and we do fewer things as we get older. And that taps into the idea that if you challenge yourself, that it helps rail against that. But specifically protective against Alzheimer's, again, you'll read 17 things to say one thing and 23 things that say something else, and so it's hard to know. I think what you've heard from all of us as non-expert experts is we're big fans of just staying healthy and keeping your brain healthy, getting sleep and exercise, and then we have to cross fingers that they're going to figure all this out before it matters for any of us. But we have no magic bullets that we know of.
Right.
So.
Okay, thanks.
All right. All right, come on. One more. Just because it's a familiar face, we'll do one more.
Well, thank you. And this is a statement, not a question. So I appreciate your end of the questions. But
Are you mad at me again?
Never.
Okay.
So I just learned 2 cures for Alzheimer's or 2 preventions just recently, and I just couldn't leave without sharing them with y'all. A young woman told me last week that shaking your head too hard causes Alzheimer's. And so I didn't want y'all to go home without that, so don't be shaking your heads. And the other one is coconut oil will cure Alzheimer's. And I just wanted y'all to thank me for those insightful presentations.
Thank you to our panelists. Laura has to leave, so thank you. Two closing remarks. 3, thank you. Thank you for being here. I hope you come again. This is our Brainstorms for April, Dr. Michael Drew. One of the things that he studies is one of the parts of the brain, one of the rare parts of the brain that actually grow new neurons as adults, and he's going to tell us about that and how it relates to things like depression, might relate to things like depression and anxiety and such. And the last thing is, so I can't help sharing this. I think it's good. I think it's good that this was a little this Brainstorms was a little confrontational, a little testy at times. This is the way scientists are. Non-scientists are always a little shocked about how scientists treat each other. We're real tough. We argue, we doubt, we play devil's advocate. And so, no, we don't. Yeah. Yes, we do. So, as a scientist, I have to say, good, healthy disagreement is healthy. And so thank you for disagreeing, and thank you for poking and pushing and asking. And I'm sorry that we didn't know answers to all the questions. But I think we should all go away and keep poking and keep questioning and doubting and pushing and find a way to get those scientists to get this done and cure it, okay? So thank you for that, and thank you for being here.